Now, finally, I want to say there's a huge explosion in longevity research with both real advances and some rather dubious claims. Now, the reason there's an explosion is because all societies are aging, and people are living longer and fertility rates are going down, with the result that the population age distribution is skewing towards older people. So it's extremely important that we find ways to have people remain healthy and productive and independent as we age. And so all developed countries are investing considerably in aging research. Of course, there's also a lot of private money with some of these motives, but not necessarily exactly the same motives. But this is the age distribution curve. This is where we are now, and you can see that it's fattening up at the older end, predicted by in 2066. And this, of course, has led to a proliferation of literature. There have been over 300,000 papers in the last 10 years. So, a book like mine, even though I worked on it for 3 years, could cover only a minuscule fraction of this research. And it's not published in just the mainstream big journals, but there is a whole proliferation of journals devoted to longevity, and new ones are cropping up every day. And along with that, there are about 700 startups which have longevity as a goal of one kind or another. And some of them will say they're really about healthy aging and not about life extension. And then there are sort of various mixtures. And there are tens of billions of dollars invested in these startups, many of them within, a few miles radius of this place. Okay. And along with that, there are also dubious claims, so this is from Tom Check, who's written a book about RNA and telomeres. And he gave me this slide, which says you can telomere shortening is considered one of the hallmarks of aging. And so there you can go on Amazon.com and you'll have lots of, things you can buy that will apparently lengthen your telomeres. But of course, these are not FDA approved. There are no clinical trials. It's not clear what their basis is. Now, at the same time, there are some promising approaches. Now, one of the things that the aging community agrees on is caloric restriction. It turns out that if you restrict animals to the bare minimum calories that they need to survive without losing weight or starving, then those animals often will have reverse many of the symptoms of aging. They will, if you look at some of the markers of aging, that will also change. And so there's a lot of effort to ask, can you mimic the effects of caloric restriction? And rapamycin, that drug I mentioned, is one such drug. But of course, rapamycin is an immunosuppressant. So you remember, it also has the potential to make you prone to infections, to wound healing. It has many other side effects as well. And people are trying to figure out what to do. Now, another interesting area that I discuss is this thing called parabiosis, where you can connect an old animal with a young animal so that their blood supplies are joined. And this means that the blood is going from the young animal into the old animal and then coming back round again. And it turns out that these animals, it's a rather grotesque experiment. And the earlier versions of this were even more grotesque, where they hadn't socialized the animal and one animal bit off the head of the other animal to which it was attached because it didn't like being attached to him. But anyway, they do things differently now. And what they've found is that the blood of the young animal helps to reverse many of the symptoms of aging in the older animal. So the older animal benefits from the young blood. And it's also true that the young animal worsens from the old blood. And this has led many labs to launch large-scale investigations into what is it about young blood that's different, and which of these factors that are different are actually beneficial, and if so, how do they act? So you can see this is a very long-term program. But that didn't stop a number of companies, as soon as these reports came out, a number of companies started up getting young donors to donate blood, extracting the plasma from them and selling it to rich people for transfusions. And, one of these companies was shut down by the FDA, and then it opened up under a different name. And then when the CEO was asked about it, he said, well, look, the trouble with clinical trials is they take too long. We don't want to wait until, for clinical trials. So that is sort of the attitude in some cases. And it hasn't stopped some people from taking matters into their own hands. And this is Brian Johnson, a tech billionaire, who decided to keep it all in the family, so to speak, because he got transfusions from his son, and he also gave blood to his father. So it's a sort of intergenerational blood transfusion progress. And this is a guy who spends, I think, about $2 million a year on longevity efforts, to keep, and his goal is to not age, and he wants to defeat aging. And, but I think he stopped the blood transfusion because he said it wasn't really actually improving his markers. But he thinks it still, ought to work. You just have to figure out, how to do it. So, and then another aspect is that many of our cells enter a stage called senescence. This happens all the time, but as we get old, we tend to accumulate senescent cells. These are a problem in themselves, but they're also a problem because they secrete inflammatory compounds, that actually are meant to attract the immune system to the site of action. And that has a deliberate purpose, but as we age, it becomes a problem. You get this large-scale systemic inflammation. And people have found that if you can specifically target senescent cells, then that does improve the symptoms of aging. So this whole field called senolytics is another big area of aging. And the final thing, which I think is most challenging, but also one of the most exciting, is something called cellular reprogramming. And we all know that, we start off from a single cell, which then divides and then eventually differentiates until a full-grown animal, the baby, is born. And during this process, the initial cell divides into two cells, which divides to a number of cells. And in the early stages, if you were to take any one of those cells, that cell has the potential to form any kind of tissue. So it's called a pluripotent stem cell because it can generate any kind of tissue. But later on, you get more specialized stem cells. For example, these hematopoietic stem cells can only generate any of the cells of the blood system. But that's a large number of cells, but still it's a limited number. For example, all of the cells of the immune system and the red blood, the precursors of the red blood cells, all come from hematopoietic stem cells. Then there are other cells which generate all of the cells of the nervous system, including neurons and glia and so on. And so as we age, both the number and the quality of stem cells decline. So we get stem cell depletion, and the remaining stem cells are what are called clonal. They're descendants from a very few stem cells, which have not necessarily been selected for, ideal properties. Rather, they're the stem cells that happen to reproduce faster. And so that is a problem. Now, people have figured out that you can take fully developed cells, differentiated cells, and actually make them go backwards in development. And these are, the most famous experiment was by a scientist named Yamanaka, who showed that only 4 factors alone could take an adult cell all the way back to pluripotent stem cells. Now, of course, if you try to grow these pluripotent stem cells, you often get teratomas, which are cancerous tumors. So it's not, there is a cancer risk with these things. And there is also the problem that if you, so to get around this, people are trying to reprogram it so these cells go backwards only a little bit and not all the way back to pluripotence. And, but then, you're dealing with a population, so you're not sure that everything is just going to be going back a little. Some of the population may go back all the way, even though the treatment was transient. So this is a field that has shown some very promising results in animals. But, applying it to humans, I think, is going to take time. It's going to take time to establish safety, long-term effects, all of these things. So I think I'll stop there, and maybe we can have a conversation.
Why we die, it's a little bit discouraging, let's face it. But I still have the same question. In the age of molecular biology, gene editing, genetic engineering, do you think that it is possible to stall aging, that immortality is within reach? Because, as you say, evolution didn't plan for death. It's just sort of an accident. So maybe we can solve it.
I think that is the feeling of what I would call the extreme optimists in the field. I think there's no physical or chemical law that says you have to die after a fixed amount of time. For example, we know that species, even vertebrates, can live over 400 years. Now, if you look at the metabolism of these species, it's very different from ours. Many of them have very slow metabolism. They're quite different. So the question is whether you can tackle the aging processes and still keep us as who we are as humans, and whether then we can do that in a safe and effective way. The question of whether we should is a whole different thing, which is philosophical. But I think it's theoretically possible, but I would put it on the same category as there's no physical or chemical law saying we can't colonize other galaxies or outer space or even Mars, and I would put it on sort of that level, and it would require huge breakthroughs, which we haven't made yet.
Right. But why is it a hard problem? I mean, you brought up embryonic stem cells. I mostly write about embryos and CRISPR babies and stuff. And so I have become very aware that immortality is all around us, because we may die, but cellular life, barring an asteroid strike, is not going to stop. So the immortality is right here, right before us. And, Dr. Yamanaka even knows the 4 factors to endow a cell with that. So why is it that we can't extract that from?
Sure. I think the reason is that we're a very complex system. It's unlike treating a single cell or a single line of cells like germ cells, which have evolved mechanisms to minimize damage. But I also think germ cells do age, but what we're doing is combining efficient mechanisms of repair so that they don't age as fast as somatic cells with a brutal selection process. I mean, what people don't realize is that, first of all, women are born with well over 1 million, germline cells. And yet, by the time of menopause, it's declined to essentially 0. And that's because it's brutal selection in the process of going from the germline precursor to the actual, egg that's, ready for fertilization in each menstrual cycle. So, it's not clear that you can do that with a somatic body. We're not, able to apply that sort of brutal selection process. And if we were able to somehow delay aging or reversing, you'd have to do that in a coherent way. So all of the systems in the body are able to do that. And then there's one final frontier where you have a brain where regeneration doesn't actually occur, and naturally. And if you regenerate, would you be able to regenerate all of the states and connections that make the brain what it is? The memory, the cognition, etc., that the brain has. That's really a sort of unsolved problem. And so I think, yes, you can do it for single cells. You might be able to do it for simple tissues even. To do it for the whole body over a very long period, I think could be a real challenge. But I agree that, there are optimists in the field, some, again, not very far from here, who, think this is a solvable problem.
Well,